Stem cells are the molecular repair workhorses of the human body. They're the engines that create, replenish and repair all of the body's organs. When a human egg and sperm join, a zygote is formed containing 23 chromosomes from each parent. The fertilized egg is totipotent. It has the potential to create an entire organism. 4 or 5 days after fertilization, cell division produces an embryo of about 150 cells called a blastocyst. Embryonic stem cells can be derived from this blastocyst. How many different kinds of stem cells are there? Pluripotent stem cells are derived from embryos, but are not themselves embryos. They can develop into most cell types. Induced pluripotent stem cells are derived from an adult somatic cell, or a cell that's not a sperm or egg. These cells can be reprogrammed to behave like embryonic stem cells. They can be taken from a patient afflicted with a disease, corrected for defects and transplanted back into the patient's body. The use of induced pluripotent stem cells in research and therapy is not as controversial as embryonic stem cells because it does not destroy an embryo. Amniotic stem cells are found in the amniotic fluid which surrounds the fetus while it's developing and is discarded after birth. These stem cells are very active, expand extensively and do not tend to produce tumors, and research and treatments using these cells don't destroy embryos. Amniotic stem cells are multipotent, and can differentiate in cells of adipogenic, osteogenic, myogenic, endothelial, hepatic and also neuronal lines. Adult stem cells typically generate the cell type of the tissue where they reside. They are tissue-specific, and are able to differentiate into various tissue types such as skin, cartilage, cardiac tissue, nerves, muscle, and bone. Cord blood stem cells are found in the placenta and umbilical cord. They contain cells used to treat genetic disorders. Research and treatment with these cells don't involve destruction of human embryos. What can stem cells do? They can divide and renew themselves many times, reproduce in the laboratory millions of times, are unspecialized. They don't have tissue-specific structures that allow it to perform specialized functions. Stem cell treatments can regenerate organs and fight disease. They can be induced to differentiate into specific cell types required to repair damaged or destroyed cells and tissues in organs such as the gut and bone marrow. Stem cells regularly divide to repair damaged tissue. They are internal repair workhorses. With other organs like the heart and pancreas, stem cells only divide under special circumstances. Recent stem cell diabetes research has resulted in reversal of the condition. There are 3,770 U.S. Food and Drug Administration sanctioned clinical trials involving SC treatment of disease and injury, including 13 trials of spinal cord injury therapy. Stem cell potency depends on how many types of stem cells they can become. Totipotent. A fertilized egg is a totipotent that can develop into all cell types, including embryonic membranes. Pluripotent can develop into all cell types except embryonic membranes. Multipotential produces many types of a closely related family of cells. Hematopoietic blood stem cells can become red or white blood cells or platelets. Oligopotential makes a few types of cells. A lymphoid stem cell can make any of the blood cells found in the lymphatic system, T or B cells or plasma cells. Quadripotential produces 4 types of cells. Tripotential 3 types. Bipotential 2 types. Unipotential 1 cell type. Nullipotential, no cell division. What are Petri dish babies? In vitro fertilization, IVF, is when a man's sperm and the woman's egg are combined in a laboratory dish, where fertilization occurs. The resulting embryo is then transferred to the woman's womb to implant and develop naturally. Usually, 2 to 4 embryos are placed in the woman's uterus at one time. Each attempt is called a cycle. Infertility affects about 6.1 million people in the United States. About 10% of men and women of reproductive age. IVF was used successfully for the first time in the United States in 1981. More than 250,000 babies have been born since then as a result of using the in vitro fertilization technique. Ethical controversies. The ethical debate is centered on research involving the creation, usage, and destruction of human embryos, and focuses on embryonic stem cells. Not all stem cell research involves the destruction of human embryos. Research on the application amniotic and induced pluripotent stem cells in disease treatment do not involve human embryos. Opponents of embryonic stem cell research compare the destruction of an embryo to an abortion. They believe that the embryo constitutes life because it has the potential to fully develop into a human being. Those against embryonic stem cell research believe that an embryo is not equivalent to human life because it is not viable outside the womb. Supporters also contend that the societal costs of many diseases and conditions, both in monetary and human suffering, means that the ethical concerns regarding embryonic stem cells are not sufficient to warrant discontinuation of this promising therapy.